Hello students, welcome to General Biology Laboratory with me, your instructor Cynthia Sanchez. I look forward to meeting you all next week as this should be our last online session of the semester. The best way to navigate this course is to go under modules. Once under modules, we are now on module 2, cells. Let's look at the activities you have for this week. Opening up module 2, pre-lab activities, you will see that lab 2 is titled, What are the Fundamentals of Life? Read chapter 2 in the lab manual. Open lab 2 in your laboratory manual so that you can create answers as you follow along with this video tutorial. Cells are the fundamental units of life. What are cells? Cells are the smallest living entity. Every organism is made up of cells. Some organisms can exist as a single cell, while others are composed of millions of cells. Cells can vary in size, shape, and function. There are two types of cells, prokaryotic cells and eukaryotic cells. Prokaryotic cells are the oldest living cells. They are simple and not compartmentalized. Eukaryotic cells are more recent cells. They are complex and compartmentalized. What do we mean by compartmentalization? This is the separation of internal structures or organelles. The separation of structures allows for each organelle to have its own designated job. Prokaryotic cells include bacteria and archaea cells. Eukaryotic cells include animal cells, plant cells, fungi, and protists. Let's look at the diversity of eukaryotic cells. Protists are unicellular organisms. These include diatoms and euglena. Animal cells are also eukaryotic cells. Animals are multicellular. Plants are also multicellular. Fungi are unicellular and multicellular. Let's look at the differences between plant cells and animal cells. Plant cells have a cell wall surrounding the cell membrane or plasma membrane. Plant cells have what's called a central vacuole. Plant cells have chloroplasts. Plant cells do not have lysosomes or centrioles. In contrast, animal cells have lysosomes and centrioles. However, they do not have a cell wall, a central vacuole, or chloroplasts. All cells have a plasma membrane. This membrane is a bilayer of phospholipids scattered about with proteins. The outer layer regulates the exchange of substances between the cell and its surroundings. Both plant and animal cells have a plasma membrane. The inside of the cell needs to be separated from the contents outside of the cell. The word intracellular means inside of the cell. The word extracellular means outside of the cell. Cytosol is the fluid inside of the cell, whereas the cytoplasm includes the fluid and the organelles inside of the cell. The cytoplasm is a gel-like substance existing inside of the plasma membrane and outside of the nucleus. Chemical reactions occur here, and the gel-like substance allows for the suspension of organelles. The nucleus houses the genetic material, or DNA. The nucleus controls cellular activities. Both plant and animal cells have a nucleus. The mitochondria is known as the powerhouse of the cell. Mitochondria release energy from sugars in the form of ATP through a process called cellular respiration. Both plant and animal cells have mitochondria. Ribosomes. Ribosomes aid in protein synthesis. Both plant and animal cells have ribosomes. The endoplasmic reticulum, or ER. The endoplasmic reticulum transports proteins and synthesizes lipids. Both plant and animal cells have an endoplasmic reticulum. The Golgi body or Golgi apparatus. The Golgi body is the site of modification and packaging of proteins, lipids, and carbohydrates. Both plant and animal cells have Golgi bodies. Some cells have a flagellum or a cilium. These structures aid in locomotion. The cell wall. The cell wall surrounds the plasma membrane in plant cells. It is composed of carbohydrates and cellulose and provides support, rigidity, and protection to the plant. Chloroplasts acquire energy through light and convert it into chemical energy through a process known as photosynthesis. Chloroplasts are only found in plant cells. These organelles contain photosynthetic pigments, such as chlorophyll. Plant cells also have a central vacuole. The central vacuole is in charge of the storage of water, wastes, and nutrients. It also influences cell size and shape. Lysosomes. Lysosomes contain enzymes for intracellular digestion of bacteria, cellular debris, and worn-out cell parts. Centrioles. Centrioles organize part of the cytoskeleton or protein filaments found in animal cells. Only animal cells have lysosomes and vacuoles. To recap, plant cells contain a cell wall, a central vacuole, and chloroplasts, whereas animal cells contain lysosomes and centrioles. Organelles found in both animal and plant cells include the plasma membrane, the cytoplasm, the nucleus, the mitochondria, the endoplasmic reticulum, ribosomes, the Golgi body, and flagella or cilia. What about viruses? Viruses are a unique entity that at this time are not considered to be living. Viruses contain genetic material and proteins on the surface. They do not contain organelles. In order for viruses to replicate, they must infect a host cell. Then they can hijack the host machinery and replicate viral genetic material. In conclusion, cells are the smallest form of life. Cells can be classified into eukaryotic or prokaryotic cells, depending on compartmentalization. Eukaryotic cells are very diverse. Although plant and animal cells are all branched under eukaryotic cells, they have a lot of similar organelles, but also have some different organelles. Viruses are not classified as cells because they require a host cell to live. Let's look at our lab for today. For Module 2 Lab, you will provide responses for exercises 1 and 2 directly in your lab manual. There is nothing to be turned in this week online. Please bring your completed lab documents to lab with you next week to be turned in person. The live demonstrations will be substituted with images shown in this presentation. Keep in mind, the language in the worksheet might not match exactly what you are doing, and the lab has been modified slightly to accommodate the online experience. This week's lab exercise will focus on eukaryotic cells, whereas Module 3 Lab will focus on prokaryotic cells. For the pre-lab review, think about the differences between prokaryotic and eukaryotic cells. What cellular features do prokaryotic and eukaryotic cells have in common? Fungi and plants are considered eukaryotic cells. They have carbohydrate-based cell walls. In fungi, the cell wall is composed of chitin and glucans. In plants, the cell wall is composed of cellulose. Bacteria, which are prokaryotic cells, have a peptidoglycan cell wall. All cell types have a plasma membrane. All cell types contain a DNA-based genome. In bacteria, which are prokaryotic, this exists as a single circular chromosome. In eukaryotic cells, the genome is composed of linear chromosomes. All cell types have cytoplasm, and all cell types have ribosomes. Some bacteria and some eukaryotic cells have one or more flagella. Recall that prokaryotic cells do not have a nucleus or membrane-bound organelles, whereas all eukaryotic cells have a nucleus and have membrane-bound organelles, or compartmentalization. Eukaryotic cells are structurally more complex than prokaryotic cells. What is meant by compartmentalization within cells? And what advantages does compartmentalization offer? The cell itself could be considered a compartment. However, In the complex cells of eukaryotes, there are multiple internal compartments and membrane-bound organelles within the larger compartment itself. Compartmentalization in any form contributes to separation, allowing activities to proceed without interference from other activities. And as long as there is some level of regulated exchange and transport among compartments, various compartments may become highly specialized and ultimately become quite efficient at doing only a few tasks extremely well on behalf of the whole cell. A eukaryotic organism can exist as a single cell, for example, a yeast, an amoeba, or a euglena. A prokaryotic organism cannot be multicellular though. However, they may aggregate to form mats or chains. The focus of this lab is to view eukaryotic unicellular organisms, otherwise known as protists, and examine them to identify their organelles. We will be looking at Paramecium, euglena, Volvox, and the amoeba. Here is an amoeba. The amoeba moves through the use of what's called a pseudopod or false foot. Microtubules are used to push the plasma membrane forward in one direction, while microtubules are retracted at the other side for the purposes of locomotion. Here we see Paramecium. The Paramecium are actually covered with very small projections called cilia. These cilia help to propel the Paramecium through its liquid environment. Here we see Volvox. Here we see euglena. Now that you have seen these four types of protists, you should draw and label your observations on page 15 of your lab manual. Let's look at the amoeba once again. What cell structures are you able to identify? For example, in the amoeba, we can see the cell membrane. It is the flexible outer boundary, and its extensions, the pseudopodia, are clearly visible. In some amoeba, you may be able to visualize the food vacuoles. These are created by the ingestion of organic debris or an organism via phagocytosis. Many of the food vacuoles will appear green, likely due to the ingestion of a photosynthetic protist, such as an algae or plant debris. The nucleus is a large, clear, transparent oval located centrally. You may also be able to see the contractile vacuole as water and waste materials are purged from the cell. What cell structures were you able to identify in the Paramecium? In the Paramecium, the first thing you should notice is the fine cilia. The cilia are not necessarily seen as individual structures, but their uniform length and rowing motion in unison creates the illusion of a clear envelope surrounding the body. The cell membrane is clearly visible, with a protein-based layer just below the outer line of a clear envelope. Paramecium have a macronucleus. The macronucleus is a large transparent structure located centrally that controls normal cell functions. A micronucleus is responsible for cell division and is not obvious. You may have noticed a ciliated oral groove that directs food inward. Paramecium also contained multiple food vacuoles and contractile vacuoles. What structures were visible in the Volvox? The Volvox is unicellular. However, the individual cells connect to one another, forming a large spherical colony, which is capable of coordinating and dividing labor. In aggregate, they contribute to the green color of the colony as these cells have chloroplasts containing the compound chlorophyll and are photosynthetic. Each cell also has two flagella that beat in a coordinated way to propel the colony toward light when photoreceptive elements within cells are stimulated. Reproductive cells produce daughter colonies by the means of asexual reproduction that appear as large inner orbs within the parental colony. What structures are visible in the euglena? The euglena has an outer boundary that consists of the cell membrane stiffened by a thin layer of protein called the pellicle. Euglena contain green chloroplasts throughout. You may have noticed a red or gold eye spot existing near the flagellum. This eye spot is used to sense the presence of light. The nucleus and contractile vacuole in the euglena are present, however, they may be hard to see. The Volvox and the euglena have flagella. Did you notice that both Volvox and euglena contain chlorophyll? This is the pigment that appears green to our eyes. Chlorophyll is used to absorb sunlight and to convert that sunlight into energy through photosynthesis. This means that both Volvox and euglena can be categorized as autotrophs. As an autotroph, it would be advantageous to be able to detect the presence of light and the position of the body to maximize light absorption. Protists are found in aquatic habitats, ranging from mud puddles, ponds, lakes, bogs, wet soils, water pipes, and water that collects in the well of flowers and leaves. Given the habitat in which these organisms live, the environmental challenges may include water balance, escaping predators, maintaining position, moving to food-rich patches, getting enough light, ingesting enough food, and reproduction. These organisms are all challenged by the diffusion of water into their bodies. For this reason, they contain contractile vacuoles that are there to actively pump out excess water. Notice that all of the protists have a means of propulsion. This propulsion can be used for escaping predators, maintaining their position in the water, and moving to food-rich patches. Also, water rapidly absorbs light. Thus, protists have means of detection and propulsion towards light. Phagocytosis is a common means for ingesting food. We find that even the protists capable of autotrophy can also fall back on heterotrophy as needed. All of these protists have a means of asexual reproduction. Rapid reproduction can occur when conditions are optimal. However, ways of exchanging genetic material with others can also occur in Amoeba, Volvox, and Paramecium, but not euglena. At this moment, we will proceed to exercise 2, the observation of multicellular organisms. The focus of this lab is to observe an example of plant and animal cells and identify their organelles. Then, you should compare and contrast the two types of cells and try to describe the structure and function relationships between the visible structures in each type of cell. The plant cell that we will be observing today is the elodia. The animal cell that we will be looking today is human cheek cells. In the elodia, you might be able to see the microtubules, the cell wall, and the chloroplasts. You can also see the cytoplasm. In cheek cells, you should be able to identify mitochondria, the nucleus, the cytoplasm, and the cell membrane. Remember that there is nothing to be turned in online this week. Instead, please bring your papers with you to lab next week, where we will undergo Laboratory 3, researching prokaryotic cells. Thank you for watching.